to record this. If everyone hits got it, we'll know that we're on, and we do record these meetings. So if anybody misses any part of it or couldn't make the meeting tonight, they all do live on our website for back reference. If anybody missed something or they want to catch up on, you can always access it there. Again, I'm Michelle Piskin. I'm Vice President of Education and Training at Caring Kind. And I'm very happy that you all have joined us. I'm very happy, Dr. Berry, that you are here. And I'm just going to get going and introduce you in a moment. I just want to talk about, for those who don't know who we are, we've been a nonprofit organization helping those in the early stages of their memory loss journey through the end of life. We also do caregiver support, and we do supportive services to care partners. A lot of our work is made possible through grants and generous donations from individuals. We do have our big walk coming up this weekend. Join us in Central Park and help support Caring Kind. I will put a link to our website in the chat. In addition, that if we do have a helpline, if anybody needs help in your caregiver journey, needs one-on-one coaching with a social worker, we are basically a social work-based organization that outreach into all the 5 boroughs. So without further ado, I'm going to introduce Dr. Berry, but Dr. Berry, I am going to give your bio if that's okay with you. Great, thank you. I did put that aside. What did I do with that? Let's see. Here we go. So Dr. Michal Schneider-Berry is the director of the new Herbert and Jacqueline Krieger Klein Alzheimer's Research Center at the Rutgers Biomedical and Health Sciences, and a professor of neurology at the Rutgers Robert Wood Johnson Medical School. Dr. Berry did a PhD in neuropsychology in Bar-Ilan University in Israel, and moved to the Icahn School of Medicine at Mount Sinai in 2001. Where Dr. Berry was a clinician for neuroscience until moving to Rutgers in April of this year. Dr. Berry also directed the Joseph Sagal Neuroscience Center at the Sheba Medical Center in Israel since 2012. Her work focuses on the role of metabolic derangement and such as diabetes and obesity, in brain aging, cognitive decline, and dementia. Her research spans from epidemiology studies of dementia and the clinical trials based on her group's discoveries. Her scientific efforts are primarily devoted to the prevention of the disease. Dr. Berry won numerous professional awards and has over 200 peer review articles, and has been funded continuously by the U.S. National Institute of Health, Alzheimer's Association, and other prestigious foundations. What you're going to learn in this presentation is Dr. Berry is going to talk about diabetes and obesity's effect on brain aging, and ultimately increases the risk of dementia. She will also share results of new clinical trials using interventions related to diabetes. So without further ado, I'm going to turn our meeting over to you, Dr. Berry, and thank you so much for being here. We're very pleased to have you tonight.
Thank you so much. And the introduction is awesome. Thank you. So I think if I see correctly, we're not a big group, right? We're a relatively small group right now. So I ask everybody who is out there, please do stop me. We scientists love our jargon. So I very easily, as much as I try not to speak scientific jargon, I tend to fluctuate in that direction. So please feel free to stop me and say, this is something that is unclear, or any question. So I'm going to go ahead and share my presentation. Give me just a second. Yeah, share this. Make it big, nice. Can you see it?
Yes.
Yeah? Okay, great. So let me just give a short intro. What I'll do is the following. I'm going to tell you what we know generally about the relationships between diabetes and dementia. And then I'm going to start breaking it down. Within diabetes, what are different things that might, or different processes, or different circumstances, or different lifestyles that may be contributing to greater risk of dementia and to higher or faster rate of cognitive decline, to brain aging in general. So this is a long set of papers, and they just keep going. You can see that those are relatively old, but they just keep going and going. And this is a highly replicated finding that diabetes is related to a high risk of dementia, both Alzheimer's disease and vascular dementia. Now, in the past, people would say, oh, diabetes is a vascular disease, so of course it's going to make more vascular dementia. Clinically speaking, it's related also to Alzheimer's disease. And remember what I'm saying now, because soon I'm going to contradict myself. Diabetes also increases the risk of MCI, of mild cognitive impairment, and it's also associated with a faster rate of cognitive decline. So de facto, diabetes is related to poor old age cognitive outcomes. Anything that has to do with cognition, old age, diabetes is not good to. So in this context, we scientists try to understand why. Okay, we try to understand what is that diabetes does to the brain that causes cognitive impairment. So numerous studies in the past have suggested that there is a shrinkage, that there is atrophy. So something in the diabetes course affects the neurons in the brain in the brain in a way that they die. So there are some studies suggesting a small hippocampus, a smaller volume of the hippocampus. Other studies suggesting in general more cortical atrophy. But if we really look at the literature broadly, everything seems to be related to vascular impairment, including in Alzheimer's disease, by the way. So for those who are familiar, type 2 diabetes patients, they have more stroke, more lacunar infarcts, the little infarcts spread, microbleeds, white matter hyperintensities, which is like little ischemia, little dots that represent lack of oxygen in certain regions, reduced cerebral blood flow, reduced cerebrovascular activity, so the vessels are less elastic and react less well to when they need to expand and contract. And those seem to be the main reasons underlying more cognitive impairment in diabetes. So as Michelle mentioned before, I was at Mount Sinai until not too long ago, and Mount Sinai has a very, very big brain bank. So we asked the question, first to see what others said, is Alzheimer's neuropathology increased in people with diabetes? So for this slide, NP is neuritic plaques, and by neuritic plaques, I mean amyloid aggregation, and NFT is neurofibrillary tangles, and that's tau aggregation. This slide, what it essentially shows is that there is no higher levels of Alzheimer's neuropathology in diabetes. No Alzheimer's pathology, no increase in Alzheimer's pathology in diabetes. This is a very important statement, because I think it kind of gave the tone for the next 10 years of our research. We actually showed, I always make a joke that someone didn't like my political opinions because it took us 5 times to publish this paper. And it's a paper actually showing that if you really, really match Alzheimer's patients, diabetic patients to non-diabetics, those with diabetes may even have a little less Alzheimer's neuropathology, and etc. Okay, there are other studies showing the same thing. No increase in Alzheimer's neuropathology. Yes, increase in cerebrovascular disease. So I want to move on. In this context, we said, okay, so we need to dig into diabetes and understand not all diabetics have Alzheimer's disease. And I must say on a more personal level, my father and mother-in-law, they're 87 years old. They're not doing well health-wise in general, but they're both severely diabetic for many, many years, and their brain is a bonbon. In fact, my joke is that I wish my mother-in-law could forget a little bit, except that she doesn't forget anything. So we wanted to understand what within diabetes is increasing the risk for dementia. And many years ago, we started the Israel Diabetes and Cognitive Decline study. And it's a study that takes place in Israel. At this moment, for unfortunate reasons, the study is stopped until the situation there becomes a little better. And the study is a collaboration between the Mount Sinai, the Shiva Medical Center, which is a big hospital in Israel, and the Maccabi Health Services. And I'm going to pause for a second just to explain something about the medical system in Israel. It's similar to some countries in Europe, but definitely very different than what happens in the United States. So Israel has socialized medicine. What that means is that there are 4 healthcare organizations in Israel that serve the whole country. And every person can choose which one to pertain to. And the law mandates that every single citizen is covered. So there is no situation that a person doesn't have health coverage or health care coverage. Maccabi, with whom we have been working for such a long time, they have an incredible data set since 1998. So, Kaiser Permanente in California, they consult with Maccabi on how to build and maintain their data sets. So sophisticated is what Maccabi has. What that means is that for each one of our participants in this research, we have 25 years of very detailed information. Think about it this way. It's all subsidized. You don't pay essentially for anything. You pay a monthly amount, but then you can go to any blood exams, any medications. You have to pay very little to get whichever type of medication you need. So we have all this wealth of data on each one of those participants. The study is longitudinal, so it means that we follow people over time. It has now over 1,000 people who are older adults with type 2 diabetes. They're all above the age of 65. And when we began the study, we required that everybody be cognitively normal at baseline. And I want to show you something that is interesting. When you have 25 years, in this case, we had 12 years of data. Now we have 25. But let's say 12 years of data. And this is a cholesterol, okay? So you can take every time point of every participant in our study and to see what is the trend of this over time, irrespective of cognition. And what you see clearly here, let me activate you guys so I know that you're with me. What does this mean? Can someone tell me what this graph looks like? So this is cholesterol levels of our 1,000 participants over a period of 12 months. What does this look like? Anybody with me there? If nobody wants to answer, I'll answer. So what this essentially shows is that over the years, historically, cholesterol levels go down. The reason that happens is because highly likely the doctors are medicating their patients with statins or other cholesterol lowering medications. And this is what you see, better levels of cholesterol over time. In contrast, hemoglobin A1C. Hemoglobin A1C is the marker for glycemic control. So the doctors want to know, when someone has diabetes, the doctor wants to know what is your glucose level at different time points of the day, according to what you eat, according to the amount of physical activity you do, etc. But they define if you have to take more or less medication according to this marker here, hemoglobin A1C, which is a 3-month marker of how well your cholesterol was controlled over this period of time. Any number below 7, most likely that the doctor is going to be relatively happy. Above 7, the doctors are going to begin saying, hey, wait a minute, maybe we have to do something with the medication. Interestingly, I'm going to say in parentheses that the American Diabetes Association now actually recommends a higher level of hemoglobin A1C as a cutoff for people with cognitive impairment. And this has to do with fear of having hypoglycemic episodes, and also fear of people having a little higher hemoglobin A1C because they forget taking medications. So that's something that is interesting for me as a clinical neuroscientist, that our data is feeding the clinicians into thinking how they're going to treat patients with cognitive impairment diabetes, accounting for their cognitive impairment. What this graph is showing is that in contrast to the cholesterol that people are treated and it goes down, I can assure you that people are treated here as well. And yet the trend is actually going up. What this tells us is that glycemic control is the personality of diabetes, okay? Diabetics, almost all of them, will have high cholesterol and high blood pressure. The vast majority of them will be obese. But what really marks this, how their disease is going to behave, is their glucose control. So we, with this data, we're able to ask the following question. Is there any trend, any patterns of how a person controls their sugar over time that is more related to cognition than other patterns? And using this data that we have, we were able to apply a certain statistical analysis that separated groups of people according to how their long-term glycemic control looks like. And what you see here, this is empirical, by the way. We didn't make any mumbo jumbo. We just gave data to the computer and said, tell us if there are groups of people whose glycemic control looks similar over time. And those are the groups that were identified. I remind you, 7, hemoglobin A1C 7, means that person, anything below that, that person is mostly well controlled. And you see that there are 3 such trajectories. So there are 3 groups of people whose hemoglobin A1C slowly going up, meaning it's getting worse over time. High levels of hemoglobin A1C means bad glycemic control. So their hemoglobin A1C is kind of slowly going up. But those people's cognition is actually doing okay. In fact, like normal people without diabetes. The people on the other 3 groups, especially this red one here, meaning that they have high hemoglobin A1C, in other words, poor glycemic control, and that the doctors are trying to get it down without much success, or because they have a violent disease, or because they're not taking their medications as they should. But those people actually have cognitive impairment. What this tells us is that it's not just diabetes. And remember my in-laws, right? It's not just having diabetes. It's having diabetes when your sugar is not well controlled that seem to be harming your brain. So one of she's not my student, she is now a PhD, Becky West, she came and said, I'm interested because if diabetes harms the brain or increases neurodegeneration, then what does it mean if you're a long-term diabetic or a short-term diabetic? What does it mean if you had 5 years of diabetes versus 30 years of diabetes? And then she said, but well, you keep pushing in, she tells me, you keep pushing me into looking at glycemic control. You keep saying glycemic control is a story. So what she tried to do, and I'll walk you through this, is she said, okay, let me look at 3 different groups of glycemic control. People who do very well, people who do moderately well with glycemic control, and people who do really poorly. And let's see, among those 3 groups, if there is any relationship, any link between how long you have been diabetic and how severe is your cognitive impairment. And what this graph in fact is telling you is that if you have good glycemic control, it really doesn't matter how long you have been diabetic. I mean, let's agree, all of us, being diabetic is a bad thing, better not to have diabetes. But if you have diabetes and you're well controlled, and essentially it does not affect your cognition, this is what our data is saying. In contrast, if you are not well controlled, then the longer you have been diabetic, the poorer your cognition will be. In fact, if you want an anecdote, we had a participant in New York. She was 94 years old. She was diabetic since the age of 50, except that when she got diabetic, she decided that she's going to control it very, very well. And we were still assessing her at the age of 94. She couldn't hear well, but she was amazing, and she was a swimmer. So in my office, I have a poster of her at the age of 80 and 85 and 90 and 92 with all the medals that she keeps getting in those swimming competitions. And that's a good anecdote for how someone who is diabetic but very, very well controlled can have a very long history of diabetes and still have very good cognition. So then we went on and asked the following questions. If you remember, I showed you a study that we had that most of the studies said no relationship between Alzheimer's pathology and diabetes. Diabetics do not have more Alzheimer's pathology. In our study, there was even a little less pathology. So we said, okay, maybe the medications for diabetes are improving the brain. Maybe the medications of diabetes are impeding more neuropathology of Alzheimer's disease. And here is, by the way, this chart is very similar to many countries in the world. So this is CDC, American CDC, but it's the same in Israel and very similar in certain countries in Europe. What this in fact is saying is the vast majority, 90% of diabetics are treated with medications. So we don't know when we see results about diabetes and brain pathology, we don't know if it's the disease itself or if it's the medications. So that's a question that we ask. We ask, are diabetes medications related to Alzheimer's brain pathology? And what we found, and by the way, I'm sure because everybody here is related to carrying kind, that you know what amyloid plaques and neurofibrillary tangles are. But this is just, in case you have not seen, this is a slice of a human brain, and this is the amyloid plaque, and this is the neurofibrillary tangle that looks like a little fish. This one happens between the neurons. This one happens inside the neurons. In any case, when we go back to our study, what I would like to take a look at is those red bars here, okay? This is neuritic plaques, this is amyloid, and this is neurofibrillary tangles, this is tau pathology. In amyloid pathology, what we found is that people who were taking both insulin and another diabetes medication that is not insulin had less amyloid pathology in the cortex, right, the gray matter, hippocampus, and amygdala. For tangles, the results were only in the cortex, but the bottom line of this paper is there is something happening when you combine insulin with another diabetes medication that maybe is neuroprotective. So we asked ourselves then, what do these results imply? What does that mean, right? Now we have to be very careful. You know, I interview quite often to the media because Alzheimer's disease is the cause I stand behind, and anything I can do to explain and to increase awareness for this disease, I do. And every time I speak to the media, I beg them to understand that when there is a statistical relationship, it doesn't mean that A caused B. So when we see that people who took diabetes medications, insulin plus another diabetes medications, have less Alzheimer's neuropathology, it does not mean that diabetes medication wiped out the pathology. It just means that those two things have somehow something to do with each other. So we said, okay, so what could be that is linking diabetes medication to less Alzheimer's neuropathology? And the direction that we take, and please don't get nervous with the never-ending jargon that every paper has. I'll walk you through it, and do stop me if there is anything that I'm saying that is not clear enough to you. So our take, or the direction that we took, was what we call insulin signaling in the brain. So think of it this way. The brain, especially the cortex, the gray matter, is full of insulin receptors. Those insulin receptors sit both on the neurons and they also sit on the vessels in the brain. And when they work, the neurons are happy. Okay, insulin signaling in the brain is a necessary process for normal brain functioning, especially cognition. So after we published our paper on insulin and non-insulin medication, another group from Asia showed that if you put in a dish, okay, not in humans, but in a dish, if you put, I'm gonna kind of cut a little bit of, kind of get to the point of this paper. If you put together insulin with metformin, and if there is anybody diabetic in the audience, you know what metformin is a number one, is the first line of medication given to diabetics. To put those two, they degrade very effectively amyloid. Another group from Brazil showed a different direction of the same thing. They said, if you put in a dish amyloid A beta oligomers, okay, so oligomers are kind of the precursor, the earlier stage of a neuritic plaque. If you put that in a dish next to neurons that have insulin receptors, the oligomer is going to stick into the receptors and not allow insulin to come in and do all the cascade that it's supposed to do for the neurons to be happy. They put insulin in that dish and they see that insulin kind of takes away or decreases the effect of amyloid oligomers, of A beta oligomers. So the cascade of its receptor is a little better. But they also found that if you put, in addition to insulin, rosiglitazone, which is a non-insulin medication for diabetes, then the neurons essentially function normally. This shows to us or suggests to us that there is something very compelling that is happening when you put together insulin and a non-insulin medication. I think I'm going to skip this one and just very quickly, we did another study in human brain tissue, and we show again that diabetes medications seem to be neuroprotective, but that most of the protection actually happens in the vessel, which was a very exciting finding for us. And from your point of view, maybe what is interesting for you to know is that the reason why we were able to do this study is that technology keeps progressing and progressing. And with time, we're able to isolate different types of cells in the brain and ask the same question that we ask in the mix of brain tissue, now each cell separately. Eventually, when we develop medications based on insulin signaling, we need to know if the medications are going to primarily affect the vessels, primarily affect the neurons, or both. I like to call research from bed to bench to bed. And what I mean by that is the following. We have an observation, all right? Seeing that in amyloid levels are lower in people who have a combination of insulin with another diabetes medication. We go to an animal model, which is this study, okay? We go to an animal model and we test it in the animal model. It's called translating back. And we find support for our hypothesis that the combination of insulin with another medication might be good to the brain. And then the next step is what we call translating back to human. Right, so it's forward translation. And at this moment, we are starting a clinical trial that was funded by the Alzheimer's Association by their part, the cloud system, where we're going to test the combination of intranasal insulin. So we're going to splash insulin into the brain so it doesn't affect levels of glucose in the periphery in the body. We're going to mix it with semaglutide, which is another diabetes medication. People know it as Ozempic, right? The injection that people take to lose weight. And we're going to focus on people with mild cognitive impairment and that have the metabolic syndrome. The metabolic syndrome is essentially prediabetes. And the reason why we want to focus on prediabetics is because they have a lot of vascular or higher levels of vascular pathology in their brain. So our chances of making a difference for them or giving a benefit to them through this combination is, in our hypothesis, higher. So if you guys invite me 3 years from now again, I'm going to have an answer if this has worked. So I'm going to move on to a different topic. Let me stop for a moment. Any questions so far? I don't see you guys, so I'm going to stay quiet for 10 seconds to see. All right. Nobody? I get offended.
If anyone has any questions, please feel free to take yourself off mute and just go ahead and ask.
I'll go ahead. Yeah? All right. So I'm going to talk to you about a completely different, not a completely different, everything is within diabetes and metabolism, but a topic that we have evolved into, and I'm actually very excited about it because I think it has therapeutic potential. So when you see me saying AGEs is not age. It's not like age 50 or age 90. AGEs is advanced glycation end products, AGEs, okay? And AGEs are a very complex chemical reaction. And this is kind of an example of what it is, that ends up in those advanced glycation end products. And I'm not going to, you know what, I don't think you're going to be interested in those details, so I'm going to go ahead. Those AGEs, they have two ways that they function. They have a receptor that if they stick to it, it begins a cascade of bad things: inflammation, oxidative stress, peripherally, and likely in the brain as well. The second way that those AGEs have their biological effects or health effects is through what we call protein crosslink. And they do cross-link amyloid. Now think there is amyloid, and there are factors that could break it, but then AGs come and link to it and don't let it break, right? So this is one of the reasons why we think diabetics might have more dementia, because they don't have more amyloid, but they can't degrade what they have. Now here's the interesting piece of it, right? AGs are primarily produced in the body endogenously, simply age and life makes more of them in your body. But they also exist naturally in foods. In parentheses, because the chemistry of AGEs is such that it glues to sugars, and this is why diabetics have very high levels of AGEs, because they have more sugar, more glucose in their blood. Those AGEs are formed with that glucose, and they have high levels. But irrespective of diabetes, AGEs are naturally present in foods, all right? So you see all this yummy here on your right? All this yummy has a lot of AGEs, right? And how are they formed? They're formed primarily in foods that are cooked in very high temperatures, and that the duration of the cooking is very long, and when there is no moisture, when there is moisture, there is less AGEs. I'm going to give you just some examples. Jaime Ribari, my friend and colleague, he did an incredible amount of work that I would have never had the guts to do. He took 750 foods, cooked them in different ways, right? Broiling, stewing, etc. And he measured chemically the amount of AGEs that there is in each one of those foods. So here you have, just as an example, that if you boil chicken, then you have about 1,000 AGEs. But if you broil it, right, make it crusty and browny, then it's 6 times higher. I think the most painful one is salmon. So raw salmon has barely AGEs, but if you broil it, then you have 8 times more AGEs than if you just eat it raw. So then a whole series of studies started happening primarily in animal models, okay? And in animal models, if you feed animals with a high AGE diet, and all you have to do, you take their food and you heat it in higher temperatures, you keep all the nutrients the same, but there is more AGEs because of the high temperature of cooking, then those animals develop more type 1 and type 2 diabetes, vascular disease, kidney disease, insulin resistance. They have a very hard time of healing diabetic wounds. And Wei Jing-Kine, my colleague, had a long study showing that they also lose lifespan, they have higher mortality. And we asked the question, do those AGEs affect dementia risk? And what we showed, I'm going to show you a series of studies until we get to the clinical trial, is that if you measure AGEs in the blood of older adults, those who have higher levels of AGEs have a faster rate of cognitive decline. With Helen Vlasaro, who's really the mother of this whole literature, we asked the question of, she developed a dietary AGEs questionnaire. And we asked the question, if people report eating higher levels of AGEs, is that related to their cognition? The answer is yes. The higher the levels of AGEs people reported they eat, the faster the decline. This was a small study. We had something like 57 or maybe 60 participants, and I'll show you a bigger study soon. So then we went ahead. Remember, we back translate. We have an observation in humans. We back translate into animal models. We want to understand, is there a mechanism behind this? When it's very well controlled, do we still see this interesting observation? And the answer, once again, is yes. You should focus on the the up red line here. Those are Alzheimer's-like mice that were fed AGEs, high levels of AGEs. When you see lines going down, it means those mice were learning, okay? It took them faster to find where they had to go because they learned. What you see in this red line at the top are that the animals that had, transgenic animals receiving out a high AGE diet, they actually don't learn, right? So it affects their brain deleteriously. This is another study that is more or less the same, and just to summarize, the same story, okay? Animals on high AGE diet did less well, but those guys were able to do, we were part of this study, but what Wei Jing, who ran this study, was able to do, she was able to measure in mice the levels of AGEs in their brain and the levels of their AGEs in the blood. And she showed that those two are very nicely correlated, right? So remember, food gets through the mouth, doesn't go to the brain. It goes to the intestines, to the stomach. Many processes happen there. And something eventually, supposedly, goes into the brain. Clearly, there is a relationship between the levels of AGEs in mice, in blood, and in brain. So then we went to the brain bank and we said, hey, let's take brains of people who have or don't have diabetes and who have or don't have a Alzheimer's disease. Let's measure their AGE levels in the brain and in the blood. And to make a long story short, what these graphs are telling you is that in humans, before I showed you in mice, in human brain tissue, the levels of AGEs are very highly correlated with the levels of AGEs in the blood. That tells me that we're taking a good direction in terms of something that we should modify to decrease the risk of Alzheimer's disease. Here is just to show you that both in brain and in blood, those who had the lowest levels of AGEs were those who did not have neither diabetes nor Alzheimer's disease, and those who had the highest levels of AGEs had both. And I mentioned before that we had another study. This is a study with over 650 people. And we show there once again that people who reported higher levels of AGEs intake in food, they had a faster rate of cognitive decline. So then from bed to bench to bed, right? We had an observation in humans, we took it back to animals, and then we wanted to do a clinical trial. And I'll tell you what we found. Ronnie Dotan, she was an excellent PhD student, really wonderful. So she ran this clinical trial. She is a nutritionist with a background in clinical trials. So she took 75 people. About half of them received a low AGE diet, and the other half received an American Diabetes Association typical diet for diabetics in general. What she did is to educate all the participants of a good diet, but in the group of low AGE diet, she also took each person's typical diet, because she didn't want to, if a person loves meat, she didn't want to say, hey, you can't have meat anymore. So she just taught them how to cook in a way that their meat would generate less AGEs. Some of the results are awesome. Some of the results are somewhat disappointing. So the first thing that she shows, and it is important to address it, is that if you eat less AGEs, the AGE levels in your blood will go down. All right. So your food affects the levels of your AGEs in the blood. And I remind you what I showed you before, both in animals and humans. High AGEs in the blood are reflected in high AGEs in the brain. So this is an important factor. If you decrease the levels of AGEs, you might, your intake of AGEs, you decrease their levels in blood and may eventually decrease their levels in the brain. With cognition and with cerebral blood flow, which was our biological marker that we were interested in, actually, we didn't find much. So let me explain to you what we found and what we think that happened, all right? And this is why I call it a bit of an ouch. So if you looked at the levels at baseline of AGEs, before the intervention, they were correlated both with cognition and with cerebral blood flow. But when we intervened over 6 months, if they had a low AGE diet or not, it didn't make much of a difference. Now we could say, okay, it doesn't work. But I don't think that's the case. I think we should give this a broader chance. I think what happens there is that when you look at the baseline level, what does it reflect? Before you begin with the intervention, it reflects your long-term lifestyle up to that point. So your long-term lifestyle reflects, if it reflects high AGEs, it shows also lower cognition. When you intervene in a 6-month period, it's too short. And if you look at the clinical trials with nutrition today, the majority are running between 3 and 5 years. And only, you need to feel pain, right? My son always says, it's a shame that when we have pain, it's when we learn the most. So we had the pain of not finding any effects of AGEs, although we have all the biology supporting that if it was a long-term clinical trial, we might have had a signal. So I'm not giving up on it. And we're going to continue trying to get funding to be able to do a long-term study. So let me stop for a moment. I'm going to stop sharing for a second, because I am all the time. Wait, here, stop share.
I think we have some questions in the chat here.
All right. So,
I
Sarah,
can read
I
them.
can't
Yeah.
believe you're on. Oh my God. There is someone I know, and I haven't spoken to her for a long time, except for recent emails for bad reasons. Yeah, so go ahead. I'm happy to answer questions, and then I'll continue.
Yeah, so I think Leticia asked, can you explain what AGE is again?
So AGEs is advanced glycation end products, okay? So think of it this way, because soon I'm going to talk about it. Everybody has thousands of proteins in their body, and those proteins have several processes that happen around it, okay? So maybe you guys heard about proteomics. So what proteomics does is to measure in blood, in brain, in urine, in saliva, the levels of those proteins. Advanced glycation end products are actually processes that happen on the protein and change the way the protein is going to express itself and function. It is a bad thing, all right? High AGEs have been related to everything that I showed you before: type 1 diabetes, type 2 diabetes, atherosclerosis, insulin resistance, etc. Is this helpful? Who was asking me before?
Hi, hello. I'm sorry. This is Letitia. I had asked a question about that.
Okay.
Then you had also mentioned salmon, and I wasn't sure if I guess salmon and then the way certain foods are cooked that could increase the AGEs.
Right.
Did I catch that correctly?
You actually you caught it very well. Yeah. So what I said is that any food that you cook with high temperatures will generate more AGEs than if you didn't cook it in high temperatures or left it raw. Then usually what people ask me at this point is, how about countries that eat a lot of vegetarian or, let's say, raw fish? Japan, for example, do they have less dementia? And unfortunately, life is complicated, but AGEs contribute a certain amount to the dementia risk. But in Japan, for example, now it's getting better, but there is endless smoking. Smoking has a lot of this, the tobacco has a lot of AGEs in it. So even though they supposedly eat less fish and more raw food, and their intake of AGEs is relatively low, they get AGEs from other sources. Not to say that smoking by itself, irrespective of AGEs, is associated with increased risk of dementia.
Thank you.
Sure. Any other questions? All right. So I'm going to share again, and I hope the next few slides are going to be I'm going to try to break it down, okay? Because for me, when I got into this new area that we call glycoproteomics, it took me forever to get it because I'm not a biologist. But from my point of view, I thought it will be important for me to share this with you because we feel we're inventing a new field, okay? We feel that we're bringing to Alzheimer's disease a new direction of thinking treatment. And I'm going to share now. Let's see. Can you see well? Olivia, make a yes. Yeah. All right. So we call ourselves the Glyco team. All right. And the leader is actually, this is a collaboration with the Weizmann Institute in Israel. So just so people, because we are American centric, but just so you know that the Weizmann Institute in Israel, the Mount Sinai, for example, in New York, has no Nobel Prize. The Weizmann Institute has 5 or 6, and then another 10 of people who were educated there, and they got the Nobel Prizes here in the United States. So that's the level of that institution. And Dr. Ishai Levin, he is the head of the proteomics in Weizmann, and David Morgenstern is a chemist, and he speaks chemistry. He doesn't speak any other language. Hila is a technician, and Sigalit is actually a lovely story. She was an acupuncturist until the age of 38, and then her mother developed Alzheimer's disease. And she was very connected to this mom, and she woke up at the age of 40 and said, if I'm not a physician who can contribute directly to Alzheimer's disease research and care, I am going to be sick for the rest of my life. At the age of 40, she started studying medicine, and she finished successfully at the age of 50, and she is now finishing her residency in neuropathology, and she is building a new brain repository. Quite remarkable. Itzik is a neurobiologist, and Nili is actually a bioinformatician, is someone who takes huge amounts of money, and I'm sorry, huge amount of money is a good thing, huge amounts of data, and makes sense of it. And just to make it interesting anecdotally for you, Neely is actually a dentist, and she got fed up of being a dentist because it was boring. And she went and did a PhD in bioinformatics, and we worked on this project together. What those people did was to develop a new technology that can take, remember, I keep saying that glucose is the whole story in diabetes, right? So they take glycan that sits on a protein, and usually the technology, let me see, I think I have a figure here. Let me see. So don't get nervous about this figure. It made my life much clearer. Hopefully I'm gonna help you understand it as well. So this is a little piece of a protein, okay? This chain is a little piece of a protein. On the protein, as I said, many things get glued to it. One of the things that get glued to it are glycans, okay? What eventually composes a glucose. Those glycans have endless forms, endless shapes, endless little pieces or components that make it have one shape and not the other. This piece of a protein with glycans on it are called glycopeptiforms, all right? Glycopeptiforms, what are they? Until now, people did proteomics, which means you take the protein, you take the little string, take away all the glycans from it, and you study the string, the protein. Others did glycomics. They cut the glycan and study the glycan irrespective of the protein. And what our group was able to do is actually to develop a technology that cuts the string with the glycan. Now, why is that important? Because the combination of those two is what defines how this protein is going to function in the body and in the brain. All right. And now I'm going to walk you through some data. I'm just trying to think what will make it interesting. I'm scared of kind of being too biological and not making it exciting for you. Although you have to believe me, it's so exciting, I can't tell you enough. Let me see. All right. I'll just tell you the story rather than explaining the graph, all right? You remember I introduced you to the Israel Diabetes and Cognitive Decline study. We took 23 people from them. 9 of them at baseline were normal. 36 months later, they were either dementing or demented already. The other 15 were normal at baseline and were absolutely normal at follow-up. We ran this glycoproteomics on them with this new technology that this group that I introduced you to developed. And we show that those who develop cognitive decline, they have different glycopeptiforms. So they have changes in their, this is in blood, by the way, in serum, in blood. They have changes in the way their glycopeptiforms work or function compared to people who maintain normal cognition. Now that is important to us in several levels, okay? This is a beginning. We're putting another grant, we're hoping to get funding to continue doing this. But why is this important? This might suggest to us that in blood, we're able to identify changes that reflect processes that are happening in the brain, all right? And it could be what we call a causal biomarker. So the blood is telling us a story about the biology of what's happening in the brain. So this is one thing that we're doing with it. But in addition, there are certain glycopeptiforms, there are certain combinations of those of the protein with the glycan that are completely novel. Nobody has tested them. So now we're moving back to the bench and running studies on animals, on mice, to try to understand if those glycopeptiforms that we see in humans that are being altered over time as cognitive decline happens, how do we maneuver them? Can we knock them out? Can we decrease their levels? Can we do something that if we move back to the human studies, we're going to be hopefully discovering a new way to change the course of decline in diabetic older adults. And this study is actually in human brain tissue. This is a collaboration with my colleagues from Rush in Chicago. And don't worry about the graph, the numbers and everything else. What we're showing here is that in the brain, there are glycopeptiforms that are related to how the person declined before death. All right. And what I want to pull your attention to is that there is one that repeats itself 3 times in different modes, but happens again and again. It's called neuronal pentraxin. So there is a neuronal pentraxin, and here's another neuronal pentraxin, and here's another neuronal pentraxin. This is new data. Okay. What this is telling us is in the brains of people for whom we have cognitive data before they died, those who had at death high levels of this neuronal pentraxin, their cognitive decline before death was very slow or none. This may be a protective glycopeptiform. So then again, we're now trying to go back into the animal models, manipulate this neuronal pentraxin, get it lower, get it higher, to try to understand if it changes mechanistically the biology of the brain of an animal that would otherwise develop Alzheimer's disease. This is down the line, but the goal is from bed to bench to bed, right? We want to go back to bed with a new type of remedies for Alzheimer's disease and cognitive decline. And I'm going to skip this. This is just, let me see if I can walk you through this. People, you remember, high neuronal pentraxin levels are related to less cognitive decline. And this is kind of a real cases. People in the low 10th percentile of neuronal pentraxin, so they have just a little bit of this good thing, their decline is faster than people in the high neuronal pentraxin levels, which is the black line. People in the 90th percentile of neuronal pentraxin, their cognitive decline was very slow. So we are very excited about this. So I'm going to conclude with with different conclusions that that hopefully summarize the huge amount of information that I threw on you right now. So the one is that a major possible reason for the discrepancy between the studies where I show you in the beginning, first, second slides, I tell you, listen, that diabetes is related with more Alzheimer's disease, right? Those are the epidemiological studies. But then the neuropathology studies say, not true. There is no more Alzheimer's neuropathology. So possibly the reason for the discrepancy is that diabetes per se is not what is causing the neurodegeneration, but it's actually specific features within it, all right? For example, patterns of glycemic control. Now, why is this important? Think of a clinical trial, okay? Think of a clinical trial on someone like my parents-in-law. They are cognitively normal, despite the fact that they are diabetic. I can tell you their glycemic control is not bad at all. They're reasonably well controlled. There's no reason to bring them to a clinical trial. They're 87, they're doing well in terms of their brain. But we know through this data that if we have a new medication, a new treatment, a new intervention for diabetes-related cognitive decline, then we should target people with poor glycemic control because we know they are the people with the highest risk of developing cognitive decline. The other thing that I showed you is that it seems that a combination of medications for diabetes, possibly through addressing different pathologies in the brain at the same time, they're neuroprotective. And we're testing this, and as I said, probably another 2-3 years, I'm going to have enough data to show. Our clinical trial for changing the levels of dietary AGE in the diet didn't show positive results for cognition. I insist, it's a bummer. No matter how we look at it, it's a bummer. I wish it wasn't. We found results already in a period of 6 months. But I think that we need to run a longer trial with older adults with diabetes, because those with diabetes have high levels of AGE, so we have an opportunity to get them down and hopefully affect their brains. And if they have high dementia risk, then we might have a chance of finding a positive effect of a low AGE diet. And first and most than everything, I thank our participants have every time again. Some of our participants, hundreds of them, in fact, they come every 18 months. We do everything. We do MRIs, PET CTs, bloods, ask them a gazillion annoying questions. And there they are, just contributing to science and to humanity. And I'm forever thankful to them. At Mount Sinai, my colleague Mary Sano, with whom I do the clinical trials, and Harry Hartunian, with whom we do the neuropathology, there are, many people are involved, obviously, but Mary and Harry, I work very closely with them. And now that I moved to Rutgers, I continue collaborating with them. And in Sheba, Ramit Ravona Springer is actually a geriatric psychiatrist that really runs the show. In Maccabi, it's a geriatrician, expert in dementia called Yossi. And then there is Itzik and Sigal running the animal studies. And this is it for me. So let me just stop sharing. And I'm happy to open the discussion to whatever questions you might have. So if people don't have questions, it can hit 2 buttons. Either it was boring, or it was so freaking clear that there are no questions.
When we think about diabetes, as we think about other diseases, high blood pressure, and other risk factors, from what you're saying, if we control our blood sugar, control our diets, that would reduce ultimately our risk factor, correct?
Well, let me say it this way.
Mm-hmm.
We don't have well, let me take it back. The answer, generally speaking, the answer is yes, okay? For every risk factor. If you look at the finger study, for example, in Finland, what they did, they controlled very well certain chronic conditions, and they pushed also more cognitive training, more social training, more physical activity. So they kind of downgraded the risk factors and enhanced the protective factors, and they did see a slow rate of cognitive decline. The issue with that, and it's a problem, is that we want a magic pill. We want to take a pill that will do like puff, and that's it. And we're not going to have any risk factors. People in general, and I'm among them. For me, I'm a chocolate addict, and I'm really making an enormous effort to decrease the amount of chocolate I'm eating. And I don't want to put the energy into it. Now, I keep thinking, what about people who are obese and love eating in general, not only chocolate? What about other bad things that we do to our health? It's very, very, very difficult to change lifestyle. And to change glycemic control, you have to change the lifestyle. Yeah, I want to mention something. Please tell me if I'm pronouncing your name correctly. Is it Laurel?
Yes.
So I read your comment right now. I'm sorry I didn't read it before, but I do want to mention to you that people with diabetes have dramatically higher levels of depression as well. So there are objective reasons, right? Because people with diabetes can't eat whatever they want. They have to be all the time tense about not having hypoglycemia and hyperglycemia. And if they have them, they feel sick. There are very big risks of losing a foot because of diabetic foot, right? Because of any injury, it's very hard for healing the wounds, etc. So there are objective reasons for diabetics to have more depression. But there is reason also to think that diabetes causes depression. I'm sorry, that depression causes diabetes. But there are people with depression that kind of let go, and then the risk for developing diabetes is higher. Now, why is this interesting in the context of what we're talking about? We have plenty of data showing that among our diabetics, those that have depression, their cognitive decline is faster. So I think your point, in addition to the personal sadness self, Laurel, it's a point that has to be well taken. If someone has diabetes and is depressed, that depression has to be well taken care of.
Here's a wonderful opportunity for anybody to ask a question. I encourage you to ask now.
Is it a problem that I made it shorter than 2 hours?
No, not at all. No, it was very rich in information.
Yeah, I'm just scared it was too rich. That's what I'm a little afraid.
It's okay. You know, it'll be posted on our website for people to review again. This is important, you know, as we look at how we can help ourselves and help our family members and understand and also enroll in clinical trials, you know, and understand the clinical trial, how those work and benefits for all those who go after us. So anybody have any other questions or that we could talk about? Okay, well, thank you, Dr. Barry, for your time tonight. Again, this will live on our website. And it'll be on demand, so people can refer to this. I know I'm going to share it with my network, as this is really important information. As you know, if we don't know somebody first degree, we know somebody second degree who has either a dementia diagnosis or dementia and diabetes diagnosis. So I do appreciate your time here tonight. Thank you so much again. Thank you everybody for being here. And I did post the Caring Kind helpline in chat. So anybody has anything they need help with, they can certainly call us for resources. We can refer them to you, Michal, if someone calls with this type of question. But thank you again for being here. We are very grateful for you volunteering your time.
Thank you. Thank you, everybody. Take care.
Thank you. Good night.
Thank you.
Good night. Bye-bye.